Why cold-blooded animals? The experiment on living tissue is usually done on a cold-blooded animal, as their survival under experimental conditions is longer than the warm-blooded animals. Among the cold-blooded animals, amphibians have long been utilized in scientific research and in education. Why frog? For studying the nerve muscle experiments, a frog is preferred animal for the following reasons: easy availability, harmless, long time in vitro tissue survival without extra oxygen and temperature control. Sciatic nerve is preferred because it is a long nerve and easy to mount. Gastrocnemius muscle is preferred as it is a bulky muscle, gives good amplitude and cannot be fatigued easily. Stunning. Hold the frog by its hind limb with a piece of cloth. Note that frog skin contains mucus glands located over the entire body. They secrete slimy mucus which protect the delicate skin from the abrasive trauma and prevent water loss. They also make frog very slippery, so frog is held with a piece of cloth. Stun the frog by its head against the edge of table. That result in unconsciousness. Feeding. Feeding of frog is done so that the animal does not feel any pain during dissection. Note that feeding of frog destroys the brain and spinal cord to abolish spinal reflexes. The muscles are flaccid because of feeding of frog, which make the dissection easier. Hold the unconscious frog in the left hand and vertiflex its head with index finger of the left hand. Feel the depression in the mid-dorsal line where it intersects the imaginary line joining the posterior border of the tympanic membranes. This indicates the junction of the skull and the vertebral column. Through this depression, insert the feeding needle and push it anteriorly into the cranial cavity and destroy the brain by rotating the needle. Next, introduce the needle into the vertebral canal and rotate it to destroy the spinal cord. Now, confirm whether brain stem is properly pithed or not by corneal reflex. Now, confirm whether spinal cord is properly pithed or not by withdrawal reflex response. Dissection. Give a circumferential skin incision at the middle of the trunk and strip the skin till the limbs. Lay the frog prone on dissecting board, cut through the muscle alongside the urostyle. Extend the incision upwards. Take care not to injure the underlying sciatic nerves. Using the scissor, cut the lower end of the urostyle and using bone cutter, cut the upper end of the urostyle and cut the vertebral column 2 cm above the origin of the sciatic plexus. Bisect the vertebral column. Remove the thigh skin and expose the sciatic nerve in the thigh. Trace its continuity proximally to the sciatic plexus and distally to the knee joint. Isolate it from the adjacent structures. Note that do not stretch the nerve or touch it with metal, can be only touched with glass seekers. Remove the skin from the gastrocnemius muscle. Tie one end of long thread around the tendon of the gastrocnemius muscle toward the muscle. Separate the gastrocnemius tendon from its attachment. Keep the length of tendon 1 cm or more. Free the muscle from its attachment. Cut the thigh and leg bone close to the knee joint. Remove redundant muscle. The muscle nerve preparation is now ready. Keep it immersed in frog ringer solution till the commencement of the experiment. Mounting. Mount the muscle nerve preparation on the myograph board. Pass a pin through the fibrous capsule of the knee joint and fix it to the cork in the board closer to the lever. Tie the tendon to the short arm of the muscle lever with a thread. Now take the record with the help of kymograph. Stimulus. A stimulus is defined as energy made free by change in the environment. There are different types of stimuli. They are physical stimulus, mechanical stimulus, chemical stimulus, and electrical stimulus. Out of the variety of stimuli mentioned, the electrical stimulus is used to stimulate the tissue for following reason. It can be localized on the tissue and applied repeatedly without producing harmful effects. Apparatus used in tidy. Therefore, the process of stimulation is easy controlled by make or break of a key or using an electronic stimulator. The intensity and duration can be easily and accurately controlled. It resembles natural mode of excitation, that is electronic and action potential. Strength of stimulus. The strength of stimulus is classified as subthreshold stimulus. It is weaker than the threshold stimulus and unable to produce a response. Threshold stimulus. It is minimal strength of stimulus to elicit a response. It is just to evoke a response. Submaximal stimulus. They are stimuli having an intensity greater than threshold and less than maximal stimuli. The response varies with the intensity of stimulus. Maximal stimulus. It is the magnitude at which a maximal response is obtained. Supramaximal stimulus. The intensity of stimulus is greater than maximal strength but does not produce an increase in response. Definition of SMT. A single action potential causes a brief contraction followed by relaxation in a muscle. This is called simple muscle twitch. The duration of a twitch vary with the type of muscle, that is fast or slow. Here is some example given from fast to slow. Correlation of simple muscle twitch and action potential on the same time scale. When the muscle action potential and simple muscle twitch are plotted on the same time scale, the twitch starts 2 milliseconds after the start of depolarization of the membrane, before repolarization is complete. Observation and results. Note that contraction does not begin immediately upon application of a stimulus. The brief interval between the moment of stimulation and the start of contraction is called latent period. It is followed by the contraction and relaxation periods. In order to calculate the duration of these periods, do not draw vertical lines from the points A, B, C and D on the time tracing. Instead, use a pair of dividers upon its point as required. Place this on the time trace and count the number of waves for each period. Calculation. Latent phase equal 0.01 into 2 equal 0.02 seconds. Contraction phase equal 0.01 into 4 equal 0.04 seconds. Relaxation phase equal 0.01 into 5 equal 0.05 seconds. Latent phase. Latent phase is the period from the point of stimulation to the starting of contraction. In this phase, no movement of liver is seen. It extends from the point of stimulus to the beginning of visible shortening. Latent phase is the time taken by the impulse to travel from the electrode along the nerve to the neuromuscular junction. Muscle excitation, contraction, coupling, and due to the inertia of liver. The duration of latent phase depends upon the length of nerve fiber that is from the point of stimulus to the neuromuscular junction and on load against which the contraction takes place. Contraction phase. Stimulation of motor nerve with threshold intensity produces conducted action potential. Release of acetylcholine into the synaptic cleft which bind with nicotinic acetylcholine receptors concentrated on motor end plate causing generation of end plate potential. Depolarization of muscle membrane by increasing its permeability to sodium ion. Generation of action potential in the muscle fiber. Inward spread of action potential along T-system. Activation of longitudinal sarcoplasmic reticulum via dihydropyridine receptors. Spread of depolarization to terminal cistern with release of calcium ion in the myofibrils via ryanodine receptors. Increase in concentration of calcium ion in intracellular fluid by 2000 times, that is from 10 to the power minus 7 moles per liter. Role of calcium. Calcium ion binds to troponin C to saturation point causing tropomyosin to move away. This exposes the binding site for myosin head on actin. Activates prosthetic group of myosin filament which acts as enzyme, that is ATPase, catalyzing the breakdown of ATP to produce energy for contraction of ectomyosin complex and activating sliding filament system. Muscular contraction. Relaxation phase. A few milliseconds after the action potential is over, the sarcoplasmic reticulum begins to reaccumulate calcium ion. The calcium ion are actively pumped by calcium magnesium ATPase, that is calcium ion pump, into longitudinal portion of the terminal cistern for storage. Once calcium ion concentration decreases in intracellular fluid sufficiently to the 10 to the power minus 7 moles per liter, the chemical interaction between myosin and actin stops and the muscle relaxes. Physiological curves. The vibration of the liver recorded after the relaxation phase are called physiological curves. Their normal duration is approximately same as duration of simple muscle twitch. They occur because of inertia of liver. Precaution in simple muscle twitch. Below are some of the precautions that need to be followed while doing simple muscle twitch. A fresh nerve muscle preparation should be used. The stimulus should be given briefly for a single induction shock. The point of stimulus should be accurately marked. Time tracing should be taken below the recordings. And muscle should not be fatigued by repeated stimulation. Effect of successive stimulus. As we know, single electrical shock of adequate strength, that is threshold, maximal or submaximal, applied to motor nerve give rise to a simple muscle twitch. If a second maximal stimulus is applied at varying intervals after the first, the response of the muscle depends upon the timing of the second stimulus. Absolute and relative refractive period. Refractive period is the brief interval of time that follows after a tissue has responded to an effective stimulus. During this time, the tissue loses its excitability, that is, it does not respond to a second stimulus. It can be divided into absolute and relative refractive periods. When the cell is refractory, the cell, that is first half of latent period, is unable to fire a second action potential, no matter how strong the second stimulus is. This is called absolute refractive period. The cause of this is that large fraction of the sodium channels are inactivated due to the first stimulus and cannot be reopened until the membrane is repolarized. If the second stimulus falls on the first half of the latent period, the arrival of the nervous impulse produces no additional response. That is, the muscle is said to be absolutely refractory. During the latter part of the action potential, that is second half of latent period of SMT, the cell is able to fire a second action potential when a stronger than the threshold stimulus is applied. This is known as a relative refractive period. It happened because some of the voltage gated sodium channels have reached their resting state. Now, if the second stimulus is applied in the second half of the latent period of SMT, the graph obtained is slightly higher in magnitude. Contraction and relaxation period. If applied later, a second muscular response is obtained, which lead to increase in height of contraction, that is summation of effects, irrespective of the phase of muscle contraction in which stimulus is applied. Beneficial effect. The contractile element in muscle do not have any refractive period, so the effect of two stimuli can sum up and give a greater response. This is called beneficial effect. The calcium ion released from the terminal cisternae during muscle contraction are pumped back into cisternae during relaxation. When the second stimulus falls just after the relaxation period, some amount of calcium is left in the sarcoplasm and it adds to the calcium released due to the second stimulus. Therefore, the height of contraction increases. The increase of temperature and decrease in viscosity due to the second stimulus also contribute to the beneficial effect. Precaution in effect of successive stimulus. Precaution to be taken while recording simple muscle twitches include the following. The point of stimulus of both the switches should be marked. Maximal stimulus should be provided so that the beneficial effect is not due to recruitment of motor units. The speed of drum should be 320 mm per second so that the graphs can be plotted on the same graph paper and can be easily compared. Nerve muscle conduction velocity. Conduction velocity of a nerve is defined as the distance traveled by the nerve impulse per unit of time. The conduction velocity varies from nerve to nerve, but as a general rule, the myelinated nerves conduct faster than the unmyelinated nerves. Two most important determinants of nerve conduction velocity are diameter of nerve fiber and myelination of nerve. In frog, the nerve conduction velocity is recorded by a simple experiment. By keeping the point of stimulation unchanged, the sciatic nerve is stimulated first near the knee joint and then at its vertebral end. The difference of the two latent periods and the length of the nerve allow calculation of the velocity of nerve impulse. Rotate the drum at the fastest speed at 640 mm per second. Stimulate the nerve at its muscle end and take a simple muscle twitch. Mark it as M curve. Also mark the point of stimulus. Keep the same baseline and without the change of point of stimulus or strength of stimulus, stimulate the nerve at its vertebral end and take another simple muscle twitch. Mark it as V curve. Take a time tracing with the help of tuning fork at frequency 100 Hz. Note the difference between the latent period of the two SMTs. Let it be t second. This represents the time taken by the impulse to travel from one point to another. Measure the distance between the central point of two pairs of electrodes. Let it be S centimeter. Calculate the conduction velocity of the nerve using the following formula. Conduction velocity equal to distance traveled in centimeter divided by time taken by nerve impulse from one point to another. Centimeter per second. Express it in meter per second. Genesis of fatigue. Fatigue. Fatigue is a word used in many different conditions. Here we concerned with muscle fatigue of the acute, objective and physical variety. So in this context, fatigue may be defined as a transient unavailability to maintain or repeat a task involving muscular contraction as a result of preceding performance of identical nature. Sites of fatigue. There are four major sites of fatigue: central nervous system, motor nerve, in the present experiment sciatic nerve, neuromuscular junction, and muscle itself, in the present experiment gastrocnemius muscle. Central nervous system and motor nerves. The onset of human fatigue is usually determined by central neural factors. Central fatigue is a failure at any link in chain of command for muscular activity that originate in the brain and traverses the spinal cord before involving the muscle. The onset of central fatigue can be delayed by adequate encouragement and motivation. However, motivation cannot influence the onset of fatigue for an indefinite period of time, indicating that there is also a peripheral site of fatigue. After the onset of fatigue, the muscle contraction can be obtained by direct stimulation of the muscle. Motor nerve is one site that has never been shown to be the site of fatigue. Neuromuscular junction. In isolated nerve muscle preparation, neuromuscular junction is the site of fatigue. The underlying cause seems to be the exhaustion of the neurotransmitter acetylcholine. It is more prominently revealed in large motor unit. Depletion of acetylcholine occurred due to prolonged high frequency stimulation. Depletion of energy stored in muscle. In intact organism, muscle seems to be the most likely site of fatigue. In prolonged work, there is correlation between fatigue and depletion of glycogen and creatine phosphate reserve in muscle, though the ATP pool is not greatly reduced so that the muscle fiber do not go into rigor. In contrast, the slow motor unit type 1 do not exhibit significant fatigue even after many hours. The decreases in force of contraction is attributed to fast motor unit type 2. Observations. Observe the change in amplitude and duration of the various phases of first three contractions and the following contractions. The first few contractions increase in amplitude due to beneficial effect. As stimulation is continued, there is a progressive increase in latent period and a decrease in amplitude. The rise of tension is slower and relaxation is more gradual and incomplete. Finally, the muscle fails to contract altogether and the lever does not return to the baseline. That is, the muscle remains in a state of partial contraction called contraction remainders. After the muscle undergoes fatigue through stimulation of its nerve, it responds briskly to direct stimulation. After a variable period, the muscle responds once again to a stimulation of its nerve. Effect of temperature on skeletal muscle. Effect of temperature. Rise in temperature within the optimal limits. Decrease the viscosity of the muscle cytoplasm. Increase the metabolic activity. Promote the activity of ion channels. The reverse effects are seen in cold environment. Effect of temperature on isotonic contraction. With increase in temperature, amplitude of isotonic shortening of muscle increases. This is due to the decrease in viscoelastic resistance to shortening and not due to increase in the number of cross bridges. The duration of different periods decreases due to changes in conduction velocity of nerve and enzymatic activity in the muscle. Effect of temperature on isometric contraction. A rise in temperature reduces the isometric twitches tension of muscle. Rise in temperature promotes the activity of calcium-magnesium ATPase, which pump back calcium faster in sarcoplasmic reticulum. Contraction is also faster due to faster diffusion of calcium from sarcoplasmic reticulum to sarcoplasm. The tension developed is also less at time for which calcium remain in sarcoplasm is less. Precautions. Below are some of the precautions that need to be followed while studying the effect of temperature on simple muscle twitch. The recording should always be taken on same baseline. Keep the point of stimulation unchanged and use the same strength of stimulus for the whole practical. The temperature of ringer solution should not exceed 42 degrees Celsius and should not be less than 5 degrees Celsius. The effect of the warm ringer should be recorded before the effect of cold ringer, since cold ringer inhibit all enzymatic activity in the muscle, which may be difficult to revert. Graph. Compare the recordings of 3 muscle twitches recorded at 3 different temperatures of ringer solution. Study the height and slope of contraction and the duration of latent period, contraction period, and relaxation period of each curve. AB1, AB2, AB3 are latent periods. B1, C1, B2, C2 and B3, C3 are contraction periods. C1, D1, C2, D2 and C3, D3 are relaxation periods. Calculation. Calculate the following time period and record your observation in a tabular format. For latent period, contraction period and relaxation period. Genesis of tetanus. Summation of contraction. If a series of maximal stimulus are applied to the motor nerve supplying the skeletal muscle, gastrocnemius in the present experiment are increasingly short interval. The contraction of muscles summate together, giving a rise to a single sustained contraction. The electrical response of a muscle fiber to repeated stimulation is like that of a nerve. The fiber is electrically refractory during the rising phase and part of falling phase of the spike potential. However, as the contractile mechanism do not have refractive period, repeated stimulation before relaxation has occurred give rise to activation of additional contractile elements. This add up to the contraction already present. This is known as summation of contraction. Increase in frequency summation. At lower frequency summation, the mechanical fusion is incomplete and the muscle give a tremulous response called incomplete tetanus. That is, there is a brief period of relaxation between contraction. The frequency of stimuli is called the subtetanic stimuli. At higher frequency, the mechanical fusion is complete and full tetanus result. That is, the muscle remain in a contracted state and does not relax. With full tetanus, it may be almost impossible to detect the slightest flicker in the mechanical record. Although discrete electrical wave can be recorded with the arrival of each nerve impulse, it is also seen that more complete the tetanus, the greater is the shortening in isotonic contraction or increased tension in case of isometric contraction. As maximal stimuli were given, the rise in tension or greater shortening cannot be attributed to more nerve and muscle fiber coming into action. Calcium level. Tension developed during tetanic contraction is about 4 times the twist tension. Tension developed during tetanic contraction is about 4 times the twist tension. When muscle is stimulated in rapid succession, calcium ion come out into the sarcoplasm with each stimulus and there is progressive accumulation of calcium in the sarcoplasm. Calcium level in the sarcoplasm reach their maximum during the SMT. The enough calcium is released in the sarcoplasm to cause complete shortening of sarcomere. However, the calcium start moving back to the sarcoplasmic reticulum before the muscle tension can reach the tetanic level. Whereas during tetanus, calcium is continuously present in the sarcoplasm and therefore the muscle get adequate time to reach tetanic contraction. Tetanic and non-tetanic contraction. Most of the muscle in the body and especially those maintaining the posture are continuously contracting. This state of contraction is not similar to tetanus. The tonic state of contraction in the body is a result of alternate successive contraction of different motor units. Tetanic contraction of certain motor units is a rare state occurring only in violent muscular effort, and such a state can last only for a very short duration. Graph. The effect of increasing frequency of stimuli on a skeletal muscle contraction, genesis of tetanus, is plotted on the graph as shown below. Staircase, trapeze, clonus, incomplete tetanus and complete tetanus. Interference. Staircase phenomenon or trapeze, it is the progressive increase in the force of contraction for a few contractions when stimulated repeatedly and when the successive stimuli fall after the relaxation period of the previous graph. If the SMT is of 100 millisecond, the frequency at which we get trapeze can be calculated by the formula 1 by 100 millisecond, that is 10 per second. Clonus. This is a state of partial tetanus observed in the experimental condition. In this, the successive stimuli falls midway of the relaxation of the previous contraction. Incomplete tetanus. This is also a state of partial tetanus where very small relaxation is obtained due to successive stimuli falling on the early phase of relaxation. Complete tetanus. It is a state of sustained contraction without any relaxation. The successive stimuli fall during the contraction phase so that the contractile elements are repeatedly activated. The individual contractions fuse together to give a single contraction.